clarification from his side. Anyway, the learning objectives, I'll just take you around the.
That's okay.
So we'll discuss the structure of sperm briefly, and then what happens from fertilization to blastocyst stage in terms of the role of sperm in that. Sperm factors, will define the sperm factors and their role in embryonic development. And ultimately, we cannot end till the time we touch upon sperm and clinical outcome. So most of the reproductive studies till now, they focus on the contribution of oocyte to embryo quality and development. And the sperm is a poor, simple subordinate. It has been always given the role of carrier of a third set of chromosomes, and the function has till now not been deciphered in clear understanding. So now the focus is changing, and people are actually beginning to read about sperm, and they want to know more about sperm and its impact on embryonic development and reproductive fitness. So human sperm are streamlined motile cells, and which are transcriptionally inert, and they consist of head, neck, and body, and a tail. The head contains a nucleus which encloses dense chromatin, and it's capped by acrosome, which has proteolytic proteins required to penetrate the zona pellucida. This lecture is quite different from what Dr. Vineet took. It's more of basic, and but then a lot of things come out of it. The nucleus is partially capped by acrosome, and the neck includes two centrioles, proximal and distal, and pericentriolar material. The tail is composed of a flagellum which propels the sperm cell towards the oocyte. This is the detailed structure. If you can see clearly, I don't have a pointer, otherwise I would explain in a better way. Take the pointer. Okay, so fertilization to the blastocyst stage. If you look at the process of fertilization, so the process of fertilization starts with the cumulus cell penetration. Obviously, there are a lot of chemotactic influences going on. Post the cumulus cell penetration, the sperm binds to the zona binding proteins. At the same time, acrosome reaction takes place. This is followed by sperm oocyte fusion, and whereby the sperm enters the oocyte, and the sperm activation, oocyte activation factor is released, which is the PLC zeta. This activates the oocyte and leads to the resumption of second meiosis, as well as cortical reaction takes place, which is instrumental in preventing the polyspermy. This is followed by the replacement of oocyte histones. They replace the protamines present in the sperm. Thus, it's known as sperm processing. Ultimately, this is followed by the PN formation. So this is all about the process of fertilization in brief. I'll go on the details as the lecture progresses. So the movement of, now the PN is formed, the movement of, if you look at the time-lapse videos, there's a typical movement of cytoplasmic material radiating from the initial position of male nucleus. This is the cytoplasmic wave-like thing which happens. It represents the initial morphokinetic manifestation of assembly of sperm tubule, sperm ester. The sperm ester is formed, and there's a cytoplasmic wave, characteristic wave, which you can see especially on time-lapse videos. And the ester enlarges from the sperm centrosome throughout the zygote cytoplasm, building the cytoskeletal infrastructure through which the PNs are repositioned. And during this PN migration, the sperm centrosome splits into two halves, whereby two daughter centrosomes are formed, and that gives rise to the formation of mitotic spindle. And this is the initiation of 2PN breakdown, and syngamy takes place. After this, in the early post-fertilization phase, the maternal derived transcripts, they regulate the initial phase of development, and there is a characteristic gene suppression as far as the sperm is concerned. At the 4 to 8 cell stage, the genetic switch happens, and the role of maternal transcripts declines, whereas a new genome becomes active. During the 3rd and 4th mitotic divisions, the process of compaction starts, whereby the inner and outer cell repositioning happens, as well as there's a formation of variety of cell junctions. And this is followed by the formation of blastocyst cavity, the inner cell mass, and the outer cell mass. So this was briefly about fertilization to the blastocyst stage. So now the question still remains, what are the sperm factors? So if you look at the structure of sperm, this is the structure of sperm. And now I'll come to the sperm factors. So there are cytoplasmic factors, genetic and epigenetic. So if you look at the cytoplasmic factors, we have got the PLC zeta and centrioles. In the genetic factors, we have got sperm, DNA, and chromosomes. And epigenetic is chromatin architecture, the methylation profile, and RNAs. So we have got experts who can comment on our sperm RNAs. Dr. Vidhu is here. She can also take a 2-hour lecture on DNA fragmentation also. So when you look at the first sperm factor, it is the sperm activating factor, or the PLC zeta. After the gametic fusion through molecular recognition and cell membrane opposition and fusion, there are periodic changes in the cytosolic concentration of calcium described as oscillations. And this leads to the oocyte activation, which leads to cortical reaction, preventing polyspermy, resumption of oocyte meiosis, selective recruitment and degradation of maternal mRNAs, and pronuclear development. This pattern of calcium peaks has an impact on early developmental competence. And when the first mitotic division begins, calcium oscillations are resumed, presumably because PLC zeta is again released in the oocyte following the breakdown of nuclear membranes. So PLC zeta not only is responsible for oocyte activation, it also has a role in the later embryonic development. In ICSI, the oocyte activation occurs only after the complete dissolution of perinuclear theca of the microinjected sperm. Centrioles are paternally inherited, and each human sperm has got proximal and distal centriole. The human oocytes also has got centriolar and PCM proteins, but they are excluded or inactivated during oogenesis. After gamete fusion, both the centrioles recruit PCM proteins and they reconstitute a centrosome to assemble the sperm ester. So this is how a sperm ester is reassembled. This is the sperm ester, which is it's like microtubules originating from centrosomes. And this sperm ester not only facilitates the repositioning of male and female pronuclei, it also prepares for the symmetric first cytokinesis. So this centriole is particularly lacking in testicular sperm, and that is why in testicular sperm you will find that a single zygote will divide directly into 3 cells most commonly, because centriole is something which is not mature in a testicular sperm. So the centrosome has a crucial role in not only in fertilization but also in the early cleavage steps. So if I look at the clinical correlations, there's an early paternal effect which emerges even before the major event of embryonic genomic activation. And there are studies which say that people have studied the quality of zygotes in a donor oocyte sharing program and in cleaving embryos developing from sibling oocytes fertilized by sperm from specific sperm donors. And they found that the fertilization from specific sperm semen samples repeatedly resulted in zygotes with abnormal pronuclear morphology, slow embryo cleavage, extensive fragmentation, and blastomeric irregularities. And they ascribed these deficiencies or discrepancies to the sperm centrosome dysfunction. Also, the centrosome dysfunctions and the consequent mitotic spindle anomalies they can induce defects in mitotic chromosomal segregation, leading to developmental anomalies. And there are studies by Tarozzi et al. In 2019, and they've reported increased proportion of mosaic embryos in severe male factor cases. And they have again said that a defective sperm centriolar apparatus is responsible for this, because it is very necessary for the fidelity of mitotic chromosome segregation during the first cell division. Now coming to the role of genetic and epigenetic sperm factors, we have got Vidhu sitting here, who is a specialist on all these things, but I'll still like to finish my part. Sperm DNA fragmentation has got many causes, and it leads to slower cell cleavage, poor day 3 embryo, and poor blastocyst development, and poor implantation. And many studies, including the study by Simone et al., which was a landmark study published in 2014, he described 3 effects: the peri-fertilization effect, which was there was a higher degree of abnormal PN when the infertility was ascribed to the oocyte. Then there was an early and late paternal effect, in which there was a higher percentage of good embryos in sperms with low DNA fragmentation and vice versa. And implantation was lower in patients who had high DNA fragmentation. So the similar findings both the speakers had found, but again they found that the implantation rate was higher following blastocyst transfer compared to early stage blastocyst transfer or late stage modular transfer. So if there is a documented case of high DFI, always go for a late blastocyst transfer. And obviously the age of female is important because the oocyte repairing mechanism is more prominent in young females compared to old females. That is the reason for higher success rates in donor cycles. If you compare it between ICSI and IVF, some authors say that ICSI works out better in cases of high DFI because of you can directly put in the sperm in the oocyte much earlier, and there is no iatrogenic damage, lab-induced damage, and you are able to select a morphologically normal sperm which is supposed to have lower DFI. But again, some authors do say that IVF does work out better because of because ICSI bypasses the natural sperm selection associated with cumulus cell vestments, oocyte vestments, allowing sperm with fragmented DNA and low reproductive fitness to contribute to the make of the embryo. So again, there are two theories, but again, if you look at the meta-analysis, it goes in favor of ICSI only. So in case there is an established high DFI case, I will prefer to go for ICSI. Sperm aneuploidy, majority of aneuploidies are due to errors in maternal meiosis. Very small proportion of embryonic chromosomal anomalies are mitotic born. If you look at the male gamete anomalies, aneuploidies, 70 to 80% of X monosomy, 50% of 47XXY, and almost 100% of 47XYY are paternal in origin, whereas almost 70 to 100% of trisomy X and autosomal trisomies are maternal origin. So why is oocyte aneuploidy more compared to sperm? Oocyte aneuploidy contributes around 15 to 20%, whereas sperm contributes 4.5%. This is due to a more efficient male meiotic checkpoint system. During spermatogenesis, the control mechanisms often cause partial or complete meiotic arrest, which leads to oligospermia or azoospermia. And there is an increase in aneuploidy rate and sex chromosome anomalies in subjects characterized by suboptimal sperm parameters. Whereas this is the reason which contributes to a higher implantation failure, pregnancy loss, recurrent spontaneous abortion, and treatment failure in ICSI cases compared to IVF cases. Sperm epigenetic factors, which is also known as soft inheritance, because it doesn't change the gene structure, but again, it involves mitotically or meiotically heritable changes in gene function without alteration in the gene sequence. It consists of sperm methylation profile, protermination of sperm chromatin, chemical modification of retained histones, and complex population of RNAs. So if you look at the epigenetics of inheritance, after fertilization, extensive demethylation occurs in the paternal and maternal genome, but there are distinct regions in paternal genome that escape demethylation. There are altered patterns of sperm DNA methylation, which were reported in oligospermic and infertile men. Sperm DNA methylation patterns are predictive of embryo quality in IVF, and genome-wide methylation patterns of men producing poor quality embryos were inherently different. So a different kind of methylation profile has been found in infertile men. Many of these abnormalities lack any clinical significance because they are assumed to be repaired during fertilization and post-fertilization development. Sperm RNAs, again, I actually read a lot about sperm RNAs for the first time. Before that, I heard it from Dr. Rajinder and Dr. Rima only. Human spermatozoa are transcriptionally and translationally inert, at least at the nuclear level. And most of the sperm RNAs are lost during cytoplasmic disposal during spermiogenesis. Majority of sperm RNAs are residual populations progressively eliminated during spermatogenesis and sperm maturation process, and they can be considered as biomarkers of male infertility without any post-testicular role. However, a complex population of RNAs, including mRNAs, non-coding RNAs, and microRNAs, are non-randomly preserved in mature spermatozoa and transferred to oocyte during fertilization. it was observed that embryos derived from sperm with altered RNA profiles had a reduced developmental competence. However, they could be rescued by injection of specific mRNAs. Obviously, these are all animal studies which we're talking about. And recently, in mice studies, we have found the authors found a decreased blastulation and live birth rate when fertilization was achieved by injecting oocytes with sperm with 90% less RNA than controls. So this just emphasizes the role of RNAs, sperm RNAs. So all these findings suggest that sperm RNAs have a potential role in early zygotic and embryonic development, though human studies obviously are lacking till now. So if you look at this is the final topic, sperm and clinical outcome. The contribution of sperm to the formation of viable embryo and its implantation, the establishment of a viable pregnancy goes beyond fertilization, as we have seen till now. The studies have suggested that pre-wash total sperm count has a positive impact on cumulative live birth rates, especially in cycles with few retrieved oocytes compared to normal or high number of oocytes. Sperm morphology has emerged as the best predictor of fertilization potential, and there are studies which say that poor sperm quality and sperm morphology are associated with higher incidence of aneuploid embryos. And obviously, if you inject poor sperm morphology sperm, then there is a decreased fertilization and implantation rate. And any sperm morphology above 6% has been shown to lead to a higher rate of implantation, pregnancy, and live birth rate. So these are pretty obvious studies, but again, the authors have taken the pain to gather the data and publish them. Advanced paternal age is associated with decreased semen volume, decreased total sperm count, and progressive motility, and as well as with increased abnormal forms, increased sperm DNA damage, altered sperm protamination, and altered semen mRNA profiles, and also the risk of spontaneous miscarriage. So this is again a topic which is very important, sources of spermatozoa and their influence on embryonic development. So according to the sperm origin, the fertilization rates are highest with ejaculated sperm, which is obvious, followed by epididymal and testicular sperms. If you take the case of testicular sperm, at the zygote stage, there is an earlier second polar body extrusion, there is a delay in pronuclei appearance, and a longer pronuclear stage is observed. And the reason for this is the oocyte does not need to substitute protamines with histones or to repair fragmented DNA sperm. So the DNA is not, it's mainly histones, and the oocyte doesn't have to replace it with, there is no sperm processing happening in short. At the cleavage stage, the embryos reach 3, 5, 7, 8, 9 stages earlier in case of testicular sperm, and the morula and blastocyst stage are reached later. If you take the case of epididymal sperm, the kinetics are similar to ejaculate, except that the blastocyst stage in epididymal sperm is also reached a little later. A higher percentage of unequal cleavage from one cell to the 3-cell stage are observed, as I explained before, in testicular sperm. This is because of incomplete maturation of centriole in the testicular sperm. Studies have also shown that a higher incidence of chromosomal anomalies in testicular sperm from non-obstructive azoospermia compared to obstructive cases, and hence the lower embryo development rates in NOA cases. Okay, what about the cryopreserved sperm? There is a decreased level of sperm mRNA in cryopreserved sperms. But again, the authors have found comparable developmental and kinetics compared to that originating from freshly ejaculated sperm. The adverse effects of sperm cryopreservation are only the loss of sperm motility and viability, and sperm vitrification protocol is epigenetically safe and induces minor biological changes compared to conventional freezing. Also, I would like to bring out this topic, this point here, because I've seen a lot of people doing a fresh TISA and never preserving, and risking the cycle cancellation also, in case there's no retrieval. Though there's a lot of publication, and even in our experience, we've always used cryopreserved sperm. We have practically never used fresh sperm. The cryopreservation of testicular or epididymal sperm in patients with obstructive or non-obstructive azoospermia is found to have no impact on ICSI outcomes and embryo development compared to fresh epididymal and testicular samples. So if I have to recap, the cytoplasmic factors are PLC zeta, which contributes to oocyte activation, centrioles, which again is important for mitotic fidelity as well as organization of oocyte centrosome and sperm ester. Genetic factors, sperm DNA, a lot has been said about DNA fragmentation and the embryonic aneuploidies. Amongst the epigenetic factors, we have got chromatin architecture, methylation profile, and RNAs, and they are all associated with reduced embryo morphological quality. So if I have to summarize, I would say that the role of sperm goes well beyond the reconstitution of diploidy in the conceptors, and it introduces a very specialized molecular toolbox inside the oocyte, which takes care of further cell divisions and embryonic development and competence. And I will go on to say that implantation is definitely a sperm function. It is more dependent on sperm probably compared to oocyte. The calcium oscillations have far-reaching effects affecting the growth and health of the fetus. And single or double-stranded sperm DNA can introduce the makeup of embryo corrupted genetic information. The single-stranded DNA damage can be repaired, but double-stranded cannot be repaired by the oocyte. A new awareness regarding the paternal role in development and reproduction is emerging, and this casts very exciting developments for male infertility and reproductive health, especially in the field of ART. Thank you.